Thank you very much for coming to everyone. I'm going to do my PhD thesis defense. Thank you very much to the committee members for coming, because I know that your agendas are really complicated and really busy, so thank you very much. And this is the title of my PhD. Thesis is Spatio-temporal processing algorithms of image sequences for automatic recognition and registration of motion. And I would like to start my presentation with a question. So the important thing here is there enough spatio-temporal information in a video to acquire knowledge about different movements? So in image sequences, you have all the spatio-temporal information to get patterns and to get information about the movements or the motion artifacts that you have there. So the important thing here, the hypothesis that we did, is to what extent can a transient motion field be captured, represented, and encoded for some subsequent information retrieval step in real time and with a minimal computational cost. This is important thing. So we need to capture, represent, and encoding this information to get everything that we need. So for that, we need to touch different fields. It's computer vision, because we need pattern recognition in different things, machine learning, because we need to do classification, and of course, image processing. So computer vision, it's a very wide field that includes a lot of different tasks or small areas, like is 3D scene reconstruction, facial recognition, object recognition, tracking, motion analysis, for example. But we are going to work in only one small area that it's a hard problem, that is spatio-temporal analysis. This is the important thing in my thesis. And of course, it includes different tasks, like, for example, we work something in content-based video retrieval, in human action recognition, of course, and in pattern recognition to the other part of my thesis. So the question here is, why is the spatio-temporal phenomena so important? Because in the image sequences, because a video is an image sequences, you have all the information included in those pixels, because you have the spatial information, because you have a distribution of pixels in your images, and you have the temporal information, because these pixels are related with the pixels in the next frame. So this is the temporal information. And you can sometimes you need to try to identify big scale objects, for example, in human action recognition, and sometimes you are interested in small scale objects, like, for example, a cell, because you want to do tracking cells. So I divide my presentation into parts. The part one is human action recognition, and the second part is in vivo intravital stabilization. So for the first part, human action recognition, to start, we can say, okay, what can computer vision do now, and what can't? So vision-based human action recognition is the automatic process of labeling image sequences with the action labels. So for that, you have to do different things, like recognition, motion analysis, sometimes tracking your body, image registration. But the question here is, there are a lot of different jobs that they are doing that. But the question is, is it possible if you want to do more bigger applications, like, for example, scoring of sports, scoring rhythmic gymnastic movements automatically with software, or, for example, content-based video retrieval. So because when you go to I don't know, some repositories like YouTube, why is the reason? Because you have to write a word. Why is the reason? Because you don't use other video to get a search of different videos that contain the same visual information that you have. Or, for example, use your webcam, use a human action, and obtain other videos that is related with that. So I would like to do a brief overview of the state of the art, because there are different important important job. So only to talk about it only a bit. For example, the first job is 3D human body tracking with multiple cameras using particle filtering. So what is important here? Well, they use multiple cameras, so you don't have only one camera, and you have a high computational cost. You are going to obtain the fine details of these movements, but you don't need maybe sometimes you don't need everything to understand the human action that they are doing. The next one, for example, is the paper of Meads in pattern recognition. Well, it's in a conference. It's stick figure models with a non-parametric Bayesian distribution. So the important thing of this paper is that they don't need to understand, or they don't need previous information about the model that you have. So, for example, you can have a giraffe moving, or you can have a person, and you don't have to understand, you don't have to have a model before about this movement, but you have a high computational cost. Other examples, the first one here is related with Hawk detectors, histogram of oriented gradients. So they use that to try to recognize human movements. The next one, for example, is short time scale limb behavior. So you have your limbs behaviors, and then with combined with hidden Markov models, they try to obtain long time scale behaviors. But the main problem here, for example, is that of course you have to view all the information, because sometimes with only one camera you can if you have a camera here, maybe sometimes you don't see this limb, for example, in all these frames. So you are going to lose a lot of information. The question here is all this information necessary, or sometimes you can use other representations of these movements to use this information. So this is the spatio-temporal templates. Spatio-temporal templates accentuate is a new representation to accentuate your movements. So, for example, if you have frame difference, is only frame difference or silhouette. It's only you have one frame and the previous frame, and you calculate the difference. So this is a spatio-temporal template. Motion history images, for example, is the same, but you are using a more number of previous frames. Motion energy images is the same, and for example, the next paper is spatio-temporal shapes. They use 3D volumes to try to create a new representation. So perfect. So now with these spatio-temporal representations, we can accentuate your movements, but now you have to classify. You need to classify different human actions so you need machine learning. So for that, the paper of Huang in 1999, it was a really good paper, because they can recognize different subjects, different humans by their gaits, by their way they were walking. So it's really interesting, and they use dimensionality reduction techniques to create a new space and work in them, because you have to think that, for example, if we have one frame, one image with 128 pixels for width and for height, you have more or less 16,000 pixels of information. And if you have a lot of image sequences, you have a very big space, so you can work in this space. So this is the reason, because you are using dimensionality reduction techniques to work better in real time in that. So we are going to talk about this about that before. So which are the problems that we found and the solutions that we propose? The problem one is large scale attributes of the movement in traditional approaches. So the question if it's necessary to obtain the fine details of the movements or is not necessary. So we propose a new spatio-temporal template, that is MVFI, motion vector flow instance, that accentuate the velocity magnitude and the direction of the movements. And then we combine with dimensionality reduction techniques like PCA, principal component analysis, and LDA, linear discriminant analysis, that we are talking about that after. How can you retain the temporal connection in multi-class classification? This is the other problem. So we understand in different problems that in this new space you have all the information that you need to classify, to distinguish different human actions, because in this spatio-temporal phase space you have everything. All the movements have a unique trajectory in this space. So this is the reason, because we analyze local differential geometry of this curve, of these trajectories, and we comparison these properties between different curves. So the first step is create the new spatio-temporal template, MBFI, motion vector flow instant, that is based on the dense optical flow. The dense optical flow is the apparent pattern to the motion from a point of view. So, for example, in this image, for example, here you can see that you divide your image in a grid, and then you create a vector for each part of this grid with the motion that you have. So the length of the vector, it depends on the velocity of your motion, and of course the direction of the vector is the direction of the motion that you have in this movement. And then we create a box for each vector, and then we use different colors. For example, here you can see that in yellow represents, for example, here I am falling, and in yellow represents the higher velocity that you have. So for example, when you are falling, you have high velocities in your head and maybe in your upper body and in your hands, but not in your feet, for example, or in your legs. So with this representation, we are accentuating velocity, magnitude, and the direction. So this is, for example, an example of comparison with different spatio-temporal templates. We are using frame difference, that is that one. And this is motion history images, and this is our template. So you can see very well in yellow the parts more involved in your movements, because sometimes, for example, when you are walking, it's important to see how your hands or your feet are moving. So it's interesting to accentuate these parts. Other examples is in gymnastic routines, because we try to classify different human actions and different gymnastic routines, but then we wanted to classify the same human action, the same exercise routine. Try to measure the quality if you do the exercise better than me. So it's really important not only classify different actions, else you want to try to get punctuations, to try to get scores for these actions. And this is an example. Well, you can see, I will go over forward, for example, you can see very well, for example, here in the passive rotation, you can see that sometimes you don't see your body, because it's not really important in this movement, because sometimes it's like static. So you need to understand very well that the important things here in that movement is the movement of your hands. With this representation, now we need to classify. So how can we classify these human actions? So for that, first we have to do a training. We have to do a training, and we use dimensionality reduction techniques like PCA and PCA, principal component analysis, and we create this new space. So for example, here we have three different actions that we want to train, and we create this space. It's important to understand here what represents each of these points. Each point represents the difference between one frame with the mean of the rest of the frames in all the image sequences. So it's really important because it's related with the covariance matrix. Then when you have created this space, you only have to get a new video that you want to classify. You transfer with your MBFI with our spatio-temporal template, and you only project this new trajectory in that space. So for example, here you can see in black that you are projecting a new action that it's related with is one of these three. You can see very well that it's really similar, more or less, that the blue one. Of course, it's not the same because here is a stack leap, it's a jump. Of course, the two girls don't jump in the same way, but you can see that the properties of the trajectory are really similar. So in that space, you can classify very well different human actions. This is the reason, because we use dimensionality reduction techniques, because you have is constructed from the orthogonal vectors that possess the most variance between all the pixels from all the image sequences. So it's important because, for example, here you remember that, for example, if we have only one frame with 128 per width and per height, you have 60, more or less, approximately 16,000 pixels of information. And if you have a lot of image sequences, you have a very big space. And here, for example, with only 3 dimensions, because we are only using three dimensions, you can see very well that you can separate two actions, that it's walking and jogging. And if you use LDA, you can separate better, because linear discriminant analysis maximize the between class variance and minimize the within class variance. So you can see the powerful of this combination of these techniques. So this is an example, for example, walking and jogging. You can see PCA and LDA, and then you project a new trajectory, is a new person jogging, and you can see very well that it matches with the trajectory that you train with other people or whatever you want. So to analyze that, we use different public human data sets, but we create two because we wanted to control all the conditions. So this is the human action repository of human actions with different human actions. We control the frame rate, the position of the camera was static, and we create a repository of rhythmic gymnastic movements to try to score these exercises. So this is the first experimental results that we obtain. So you can see with this data set, so you can see in the first 3 figures that in black you have MBFI. So we are comparing MBFI with motion history images with frame difference, and then we calculate the recognition rate only in this new space using a simple distance metric like KNN, so K nearest neighbor. So here you can see that the recognition rate is always better with our technique instead of the number of persons that you are using in your training and the number of actions that you are training. For example, here is 2, 3, and 5. Below, in the figures below, you can see the first figure is related with the silhouette. This is motion history images, and this is MBFI. And you can see in MBF5 you always obtains better recognition rate between 85 and 98%, according to well, independently of the number of persons that we are using in the training and the number of actions that you want to classify so the next problem that we want to solve, that we want to work, it's to try to classify different gymnastic movements, but to try to score the same gymnastic movement done by different girls, by different athletes. So this is an example of 3 actions that you want to classify. So we create the motion vector flow instance, and then we create this space, and now we have a new girl doing the same action. I don't know, I don't remember, but I think it's a jump. So you can see very well that the projection in this space, it's really similar to the trajectory of the same exercise did by other girl. So here, for example, I think it's a jump, or yeah, it's a jump. So now you have a new video, your space is created. So you only have this is in real time, because you only have to get your video transfer with the motion vector flow instance, but it's in real time, it's really fast. And then you only have to project in this new space, and you can see in the yellow one that it's really similar, the distribution of the trajectory, the amplitude, with the blue one, that is the action that that you train. So the results here, I think it's really interesting, because we use different, several judges, several experts. They evaluate all the videos, and they put different scores, and then we use our software to try to get the results with our software and compare the results. So for that, we have to do a first step of calibrating. So we have a KNN weighted score, because we use in this space K nearest neighbor. So we need to use several videos with the punctuation of the experts with our software, and obtain the results with the KNN, K nearest neighbor, and calibrate the software. And then we can use new videos, and you can see very well that with new videos, you can see in blue one, the blue one is the judge evaluation, the expert evaluation, and in yellow, well, it's yellow or green more or less, is our system evaluation. So you can see very well that you can obtain the same punctuations, the same scores. You can see very well really fast, because you can see two videos, it's people doing a walkover, two girls that they have high scores by the experts. You can see very well that the trajectory are really similar, and one girl that obtained a low score, the trajectory is completely different. So you can see very well that in that space you can distinguish very well different actions, and not only different actions, else you can distinguish the quality of the same action. So here it was when we understood that it's not important to understand. It's not important to try to use a KNN, because you are losing a lot of information, because you have a lot of information in these trajectories that you are losing. So it's important to understand the local differential geometry of these trajectories, because you can have different cases that KNN work bad. Because, for example, in the first region, like here, you can see that you have two actions, two trajectories that are really overlap, because maybe it's the same trajectory. So of course they are more or less overlap. So you need to use if you use a distance metric like K nearest neighbor, it works bad, because all the points are really close. So, but you can use the local differential geometry properties, and you can create a hyperplane osculating, a mean osculating hyperplane, based on the Frenet-Surratt equation. Is based on the is normal by normal and torsion, and you can calculate very well the hyperplane, and this hyperplane is unique for each trajectory. In other cases, for example, if you have the trajectories very separated, it's not important, because the centroid of the trajectory can be useful. Or for example, in that one, you can have some part of the trajectories overlap. So in that case, maybe you are interested in using a fuzzy KNN, not only based in the distance with other points. You want to understand better the direction of this of each point in this part of the curve, in this part of the trajectory. So this is the reason, because we have a new distance metric that you can ponderate the different things in the different cases that you can have. So this is the results that we obtain. So for example, here I think it's important, you have a trajectory, you have an action that it's really overlapped with other action that it's bigger. So you can see that with KNN, K nearest neighbor, you obtain a very bad recognition rate, but well, it's not very bad, 75%. But with this new distance metrics, trajectory point cloud, you obtain 91%. So I think it's really better. And sometimes, for example, here the KNN can works well, so you obtain the same recognition rate. And we try to do content-based video retrieval, preselecting some parts of two movies. But of course, here you have a lot of false negatives and false positives, because you have a lot of problems here, because the point of view is changing a lot. You have zoom, you have maybe sometimes a lot of people in the scene, so you have a lot of problems that you need to correct, and not only with one camera and static camera, not only changing continuously between one camera and another camera. So in a movie you have a lot of problems that you need to correct with different techniques. So the conclusions and future work are related with human action recognition. So as the most salient aspects of the movements are fully contained in the spatio-temporal phase space, because each movement is represented in a unique trajectory in this space. So you can classify very well, or you can distinguish very well in this space different human actions. Well, and the pattern recognition in human actions is tantamount, is equivalent to storing, recalling, and comparing the phase space trajectories. Feature directions, multi-scale integration, multi-scale and multimodal in actions in daily life. It's related because sometimes you need to understand short time scale behaviors, like for example, you are drinking, but sometimes you have to want to understand long time scale behaviors. For example, your routine in a week, for example, in your home or something like that. So you can understand that in this space you can do new work in these fields. This is, for example, in actions in daily life, relative quality of the movement. So there are more things that you can do to understand better the local differential geometry of these curves. Because, for example, we started with a KNN and we are losing a lot of information, and then we understood that you have a lot of information in these curves that you are losing, not only with the simplest distance metrics. Well, I am extensions for content-based video retrieval related with the things that I said before, related because you have a lot of change of cameras, so you have to synchronize this information if you are looking to a person from here or from there. So it's really complicated to try to solve that. But you can do with optical flow. You can remove this. For example, if you have zoom or something like that, you can remove with optical flow, because you can see the vectors that they are pointing to a point or something like that. So it's you can remove or for example, if the camera is moving, you can remove this motion related with the camera and not with the person walking, for example. And the second part of my thesis, well, I need a bit of drink. Sorry. The second part is in vivo intravital imaging stabilization. So the important thing here is computer vision necessary in bioimaging or in vivo intravital imaging? So you have new techniques like two-photon microscope combined with intravital imaging techniques that they are really good to see tissue alive with a very good depth. But sometimes you have a lot of problems. So because with these techniques you can visualize cellular and subcellular structures, but of course the mice is alive and you have a lot of different motion artifacts, because like displacements, rotations, change of scale. The reason is because you have the respiration of the animal, you have cardiac activity. So if you want to have a very good resolution, of course you are going to obtain more noise and more motion. So you need, this is the reason, because post-processing software can improve your data. So not only the two-photon microscope combined with intravital imaging techniques, that the way that you are getting those images, you can use post-processing software to try to improve this data. So which are the problems that we found and the solutions that we propose? The problem one is the background motion of the tissue. So for that, we have a contour-weighted cross-correlation with a background movement model. We are going to talk about that after. The problem two is drifts or long time scale variations. So the stabilization is constrained globally with a Monte Carlo optimization procedure. So you don't want a drift over time, or you don't want a very big change from one frame to the next one. Problem three is intravital imaging techniques problems due to undersampling. Well, it's undersampler errors. It's related because when the microscope gets a stack of images, it's not an atomic time. You need a lot of time to get all these images. And of course, in that time, the tissue is moving. So you can have a lot of different problems. And when you obtain first slide, for example, can have some part of the information that you are obtaining in the second slice. So you need to choose one of them, for example. We are going to talk about that after. So only a brief overview about the state of the art of the main jobs that people were done. So there are a lot of physical devices that people are trying to do to try to reduce or to try to remove the motion. For example, the first one is a microstage device that reduces organ motion. It's for abdominal organs. So they try to put a physical device to try to correct or to try to remove the movement. And the second was, for example, a new water immersion suction stabilizer that using a water-based stick, and they try to reduce with this water-based stick the motion. It's like for different things. I think it's for cardiac imaging. But there are software too. There are different software tools to try to correct these motion artifacts. For example, the first one is a Fiji plugin, but it's only for 2D stabilization. Other paper, for example, in Science Reports in Nature, is removing motion artifacts based on the maximum correlations in some parts of the images. So they choose several parts of the images and they try to obtain the maximum correlations to remove some motion artifacts. So we need to understand better how the microscope obtain the images. So here, for example, is a multiphoto microscope. Is the multiphoto microscope that in the Ulrich Institute they have. So Ninka knows more than me about that. And here you put in a box your mouse, and now you are going to obtain the images. So you are going to obtain in one cycle a lot of slices with a different focal length. You are going to obtain different channels. So you repeat that in different cycles. But the main problem here is that this cycle is not an atomic time. You need a lot of time to get the first slide, maybe several seconds, maybe minutes. You need more minutes or more seconds to get the second slice. To obtain every slices, you need a lot of time. So here you can have different motion artifacts. You can have movements in XY. In XY between one slice to the next one, or movements in Z, because the stacks can be moving, or maybe both movements in XY and in Z. You can have rotation, or you can have change of scale. Well, it's not change of scale. It's more related with sometimes the tissue can be compressing, or you have a compression, or you have a stretching, and sometimes the tissue can be closer or farther to the lens. So you have like a change of scale, but it's not change of scale. Sometimes the tissue is closer or farther to the lens. So for that, if you have two images and you have a displacement and you have a motion, you can use a simple convolution. So you have one image and you have another image, and you do a convolution, you obtain the best matching. But of course, it's not the best way to do stabilization because you have a high computational cost. The more elegant solution, the more elegant and the more efficient is using Fourier transform. So if you use Fourier transform and phase correlation, you can correct movements in XY, because given two images, F1 and F2, which differ by a movement, dx dy, the main property of the Fourier transform is that you have the same Fourier magnitude, but you have a phase difference. So if you calculate this phase difference, is this one, you are going to obtain a point that represents the displacement of your images. So it's perfect to try to understand or to try to get the movements in XY. But so with this technique, you can correct movements in XY and in C, because in C you only have to compare this stack with all the combinations with the next one, obtain the best match, the best match with a distance metric that we are going to talk after. To correct rotation, you can use log polar transformation, which is the main property of log polar transformation, that the scale and rotation in the Cartesian domain is your normal domain in your images, represent a movement in XY in the log polar domain. So for example, if you have a rotation in our image, you are going to obtain a movement in Y in the polar domain. So, and if you have a change of a scale, you are going to obtain a movement in X in the polar domain. So if you combine both, you are going to obtain the information about scale and rotation. The main problem here is a simple translation, dx dy, produce a modification of the log polar image. So these equations change a lot, but the solution is work in the Fourier space, and you obtain the solution. So the main problem here, okay, if you use these techniques that they are not new, you need to try to get a measure. So how can you measure the best alignment? How can you measure the best stabilization? Because there isn't anything to try to measure that. Sometimes normally it's the expert that say, okay, this stabilization, it's really good, you have a very good you have improved data, but it's not a quantitative measure. So for that we create a pixel overlap metric based on Monte Carlo random samples. So you put a lot of random samples over your images, and you compare one random sample in one image and in the next one, and you compare by different things. But for example, here we use the histogram of color. So with this metric, you are able to compare the quality of the different corrections between two images. So when you have the best displacement corrected, you are going to obtain a very good results with this metric, with this pixel overlap metric, because you are comparing different random samples in the same position in this image and in the next one, and the matching, it's really good. So with this technique, we can measure the alignment. But the main problem here is that in this data you have a lot of movements, but sometimes it's not linear movements. You have endogenous movements, because the tissue can be you can have a compression, or the tissue can be stretching. So sometimes you have some part of the images that you don't want to use in the stabilization. This is the reason, because using the same technique of a Monte Carlo random samples, you can well, we use previous, this is the original image, we use a contour threshold. It's only to lose some pixels that you don't want to use. But then you can use these random samples to select the part of the images. This point is only the center of a random sample. The part of the images that you want to use, because here you can see, or maybe, well, you are going to see after. But here it's you can see here. Here you can see, for example, is the original image. This is the next frame, and you can see very well, like the tissue is stretching a lot. So in that case, you don't want to use this part of the image, because you are going to obtain a bad stabilization because of that. So you want to remove part of the images part of the images, and then doing the using the same techniques for your transfer, log polar transformation, and phase correlation to do the stabilization. So this is the reason because we use this technique to remove part of the images. So here we have a model for degree of nonlinearity when you have endogenous movement. When you have, for example, a compression like here, the tissue between here to here, you have a compression, you have an unimodal distribution. You have an unimodal distribution because if you have several cut planes over time, you can see that the distribution of these points in red, that is, it's random samples that they don't match very well because of that, because of the compression. You can see a unimodal distribution, and you can see when you have stretching, it's over time, it's bimodal distribution. It's like the tissue is stretching, and you have two parts in the image that they are moving a lot, but the central part is the same. So you can use the central part of this stretching to do the stabilization. This is the reason because we use this technique. Then we have a Markov chain Monte Carlo update step because well, we think that the best algorithm will do a globally consistent alignment between all image frames. So the average area for all the contours that we use, that it's this one, is this. The contours, you have to you are going to obtain the same area. For example, from one frame, from one cycle to the next one, you are not going to obtain very big difference. So, or maybe sometimes over time, you don't want to obtain a drift over time. So this is the reason because we use this technique to try to get that. And finally, the undersample errors. The undersample errors is related with the things that we talked before, related that aside all the images, all the slices that you obtain in a cycle is not in an atomic time. You need a lot of time to get the image. So, for example, this is an exaggerated example, but for example, the time that the microscope needs to obtain the first slice is the time that the tissue is moving a lot. And sometimes you are going to obtain the second slice with the same or more or less the same information that in the first one. So, for example, if you are doing cell tracking, you can get one cell in the first slice and another cell in the second slice, but they are not two different cells, they are the same. So you can answer biological questions with this data because it's wrong. You need to remove one of them if you know that they are the same information. So using the same techniques that we used before, we can remove some of the slices that you have. Well, finally, related with the results, experimental results that we obtain, we create a stock application in Python with OpenCV library, and you can see in these figures the comparison of the results as a function of time. So you can see it's like violet. It's using linear correction. Linear correction, it's only using Fourier space with log polar transformation and phase correlation. And in green, you can see using our model. So selecting the part of the images that you want to use to do the stabilization. And you can see that you have a bigger a better stabilization because these are corresponds with the overlap pixel metric. So you are measuring the quality of your stabilization. So if you have different displacements, you correct them and you use this metric to measure the quality of your stabilization, because before we didn't have anything to say if it's a good stabilization or not. Only the experts can say, okay, that is better, but with this technique we can measure that. This is a small tracking of a bit, but it's only to see that the difference of a track between in a stabilized data, you can see in blue, and the original data. So you can see that it's completely different, and it's really important to stabilize the data, because in other cases you are going to obtain different results that they are not true. Here in this figure, I think it's really interesting because we have three types of data: Phi1, Phi2, and Phi3. In Phi1, it's data with linear displacements. It's only linear displacements. Phi2, you have some part of the endogenous movement, but not a lot. And in Phi3, you have a lot of endogenous movement with compressions and stretching. So you can see in blue that we are using our techniques using our nonlinear background model. So you can see very well that you can obtain very good results, better results, that if you use only Fourier transfer with log polar transformation and phase correlation. So if you use our model, you obtain better results in this type of data. In other cases, with linear distributions, with linear movements, you are going to obtain more or less the same results. Well, this is some examples that this is, for example, this is the original video that you only have a small movement in X, Y, not a lot in Y, and you can see that the stabilized video using these techniques. So this is not very complicated because it's linear movement. You don't have a lot of endogenous movement. Other example is from data that we from Utrecht Institute from the Netherlands. So it's mammary tissue data with breast cancer, and you can see with more movement, you can see that in the original video the tissue is like moving up and to the left. You can see, for example, this structure is moving up and to the left. And here, for example, with this technique, you can see very well that you have this part of the tissue in the same point. Of course, here you have the two videos have the same resolution, but here you have a bigger background in black, and then you put the images in your displacements. In the last video that I wanted to show, I think it's really interesting. It's related with the job that we did in Utrecht in the Netherlands with the Jacob van Rennes group, and it's data from the small intestine, and it's important to try to show that because it's only a vertical stack, so it's only a stack in one cycle, and you have the problem that the microscope can get all the different channels in the same time point. So you have to do a scan and get some channels, YFP, GFP. Well, you have to obtain several channels, and then continuously you obtain the other channels in the second scan. But of course, in that time you have a different movement, and you have movements, and you can see very well here. For example, I put this square only for help, but you can see very well in gray and in red that it's not really overlap because of that, because the first scan is not corresponds with the second one. But here with our techniques, you can see very well that they are overlap, and using these techniques, using phase correlation, log polar transformation, and using our nonlinear model, you can use this transformation. In that case, it's not really important because you don't have a lot of endogenous movement. But for example, in placenta data, you have a very, very big movement. So it's really important to try to use that. And conclusions and future work, well, the connection or mapping between time points in space contains essential information of the underlying motion. So it's important to try to understand the spatio-temporal information that you obtain, that you have in these pixels of your images, and the motion field encodes details of the underlying anchors or landmarks landmarks that which can be used as well as the nonlinear background. This is the reason because we wanted to select the part of the images that that we wanted to use. The future directions, for example, in nonlinear background, it would be good to do a comparison with diffeomorphic maps, because there are different diffeomorphic mapping techniques. So it would be good to compare the results with these techniques with real diffeomorphic maps techniques. The global stabilization, should we use simple simulated annealing based on the average area, but it would be good to analyze different techniques, other techniques to try to do that. Parameter extraction of biological importance. So of course, we do that because we are interested in answer biological questions. It's the biologists or the immunologists who answer these questions, but it's important to compare the results with this data, how the things you can answer with this improved data, which are the things that you didn't answer before. So I think it's really important to show that. And general conclusions about my thesis is, okay, spatio-temporal features in video stream can be encoded so as to preserve the underlying information content. So, for example, in the human action recognition, this spatio-temporal phenomena can be represented in different spatio-temporal templates, and then in this new space you have a unique trajectory that corresponds with a unique movement. So I think it's really important to understand that. The methodology has been through practical problems, and these problems had led theoretical and broader questions. This is the reason because well, we were talking about different applications or different problems during my thesis. Several fields have been touched, of course, computer vision related with pattern recognition, machine learning related with classification, information theory, theory of computation, differential geometry of physics. And there is a unified principle between the different problems in human action recognition and in bioimaging or in vivo intravital stabilization is the way of treating the spatio-temporal field. So spatio-temporal information that you obtain and the relation between each pixels of an image with the next one. So how can you capture this information? How can you represent this information? And then how can you encode this information? Well, and finally, the acknowledgements, of course, to my family and to my friends, to my family because, to my mother and my sister because they helped me a lot during this period. Big part of this thesis is of you. My friends, because of course I have very good moments and very good opportunities to try to forget sometimes my PhD. My PhD advisors, because they helped me a lot of questions, a lot of different problems that I had. David, for example, has every day new ideas for everything. I don't know why, but but I think it's incredible. I can't do that. Of course, Van Rennes research group, because when I was working in Utrecht in the Netherlands, it was really interesting for me because I can learn a lot. It was a very good research level there. A very good environment to work. So it was really amazing for me. The same in Lisbon in Carlo Tadocoro's research group, because I started to work with biologists and try to understand different points of view of engineers. So I think it's really interesting. And I started to work with two-photon microscope and understand how they are getting the images. Well, of course, finally the funding, the Xunta de Galicia, because give me the opportunity to have a contract in this period, but now it's finished. But okay, but Fundación Barrier, because they gave me the opportunity to go to Utrecht. That I think it was really important for me. Campus do Mar, Marine Instruments and Innova, because they gave me a very important research award in this period. So, well, that is all. I would like to answer all the questions that you could have. So the question here is, why is the spatio-temporal phenomena so important? Because in the image sequences, because a video is an image sequences, you have all the information included in those pixels, because you have the spatial information, because you have a distribution of pixels in your images, and you have the temporal information, because these pixels are related with the pixels in the next frame. So this is the temporal information. And you can sometimes you need to try to identify big scale objects, for example in human action recognition, and sometimes you are interested in small scale objects, like for example a cell, because you want to do tracking cells. So I divide my presentation into parts. The part one is human action recognition, and the second part is in vivo intravital stabilization. So for the first part, human action recognition, to start we can say, okay, what can computer vision do now and what can't? So vision-based human action recognition is the automatic process of labeling image sequences with the action labels. So for that, you have to do different things like recognition, motion analysis, sometimes tracking your body, image registration. But the question here is, there are a lot of different jobs that they are doing that. But the question is, is it possible if you want to do more bigger applications, like for example, scoring of sports, scoring rhythmic gymnastic movements automatically with software, or for example, content-based video retrieval. So because when you go to I don't know, some repositories like YouTube, why is the reason? Because you have to write a word. Why is the reason? Because you don't use other video to get a search of different videos that contain the same visual information that you have. Or for example, use your webcam, use a human action, and obtain other videos that is related with that. So thank you very much for coming to everyone. I'm going to do my PhD thesis defense. Thank you very much to the committee members for coming, because I know that your agendas are really complicated and really busy. So thank you very much. And this is the title of my PhD. My PhD thesis is Spatio-temporal processing algorithms of image sequences for automatic recognition and registration of motion. And I would like to start my presentation with a question. So the important thing here is there enough spatio-temporal information in a video to acquire knowledge about different movements? So in image sequences, you have all the spatio-temporal information to get patterns and to get information about the movements or the motion artifacts that you have there. So the important thing here, the hypothesis that we did, is to what extent can a transient motion field be captured, represented, and encoded for some subsequent information retrieval step in real time and with a minimal computational cost. This is important thing. So we need to capture, representate, and encoding this information to get everything that we need. So for that, we need to touch different fields. It's computer vision, because we need pattern recognition in different things, machine learning, because we need to do classification, and of course, image processing. So computer vision, it's a very wide field that includes a lot of different tasks or small areas. like is 3D scene reconstruction, facial recognition, object recognition, tracking, motion analysis, for example. But we are going to work in only one small area that it's a hard problem, that is spatio-temporal analysis. This is the important thing in my thesis. And of course, it includes different tasks, like, for example, we work something in content-based video retrieval, in human action recognition, of course, and in pattern recognition to the other part of my thesis. So I would like to do a brief overview of the state of the art, because there are different important jobs. So only to talk about it only a bit. For example, the first job is 3D human body tracking with multiple cameras using particle filtering. So what is important here? Well, they use multiple cameras, so you don't have only one camera, and you have a high computational cost. You are going to obtain the fine details of these movements, but you don't need, maybe sometimes you don't need everything to understand the human action that they are doing. The next one, for example, is the paper of Mitch in pattern recognition. Well, it's in a conference. It's stick figure models with a non-parametric Bayesian distribution. So the important thing of this paper is that they don't need to understand, or they don't need previous information about the model that you have. So, for example, you can have a giraffe moving, or you can